Before the seminar, please allow me to introduce and also thank you very much for Pernille to give us a talk. And I will introduce her a little bit at the beginning. So she works as a service president, the head of global clinical evidence and also medical affairs for SSI Diagnostica Group. And she has been always passionate about infectious diseases. And she has more than 20 years experience in this area. That's a lot. And also, she's a member of the surveillance community and has been part of WHO reference lab as well. So today, she will give us a talk on the title of Antigen expression and antibody strategies in vaccine. QC use nephelometry. So please join me to welcome our speaker, Pernille. Yeah. Okay. I will hand over this to you. So yeah, thank you.
Thank you so much, and thank you for having me. I will now share my presentation.
Sure.
So I hope everybody now can see the screen.
Yes, I can see that.
Okay. So first of all, thank you everybody for joining the webinar, which is called Beyond Genotyping and Gene Expression and Antibody Strategies in Vaccine QC using the nephelometry method. So first, I would like to give a short introduction to myself. Thank you for the nice presentation of me. So my name is Pernille Elverdal. My educational background is that I have a master in science within the biotechnology area. I'm coming out of the Technical University of Denmark. And on top of that, I did my PhD in respiratory diseases in a specific bacteria called Legionella, which is an atypical pneumonia, where I wanted to improve the diagnostics and the surveillance of this very severe disease. And that was done in the National Reference Laboratory in Denmark. So in 2006, I started working at the Danish National Reference Laboratory called Statens Serum Institute. Here I developed new diagnostic methods, and we're part of the surveillance industry for the respiratory diseases, primarily the atypical pneumonia, but also in the pneumococcal disease area. In 2010, I changed my role. I started working in the department called SSI Diagnostica. It was still under Statens Serum Institute, the National Reference Laboratory, but here I became the specialist and the technical manager for some of our products, which was the Pneumococcus and Streptococcus and Haemophilus influenza and Legionella portfolio. But we also had at the same time portfolios within E. coli and Salmonella, which was being used for the surveillance industry. In 2016, the government decided that they wanted to prioritize SSI Diagnostica, so they sold off that department who were selling the antisera and the antigens and also commercial kits for the whole world within surveillance, but also the diagnostic area. And that was when I ended up heading the R&D department in the company, but still continuing my passion within pneumococcus as a specialist within the typing of pneumococcus, but also the support to the vaccine industry within quality control of their vaccines. And then today I'm sitting as the senior VP and heading our global clinical evidence group and medical affairs, still supporting the industry, both the vaccine industry, but also the surveillance industry and the diagnostic space. So that was shortly about myself. And now I want to just give you a short introduction because I'm going to cover a lot during this presentation. Seeing if I can move it over here. So the agenda for today is that I actually want to show you things which you need to be aware of when you're developing vaccines, and here specifically the bacterial vaccines. I will give you, as my background is coming out of the pneumococcus space, I will give you a short background history lesson on pneumococcal serotyping and its surveillance, and also the vaccine development being done in that space. I will then move into the phenotypic method, why that's important and critical during the vaccine development and in the surveillance. And then finally, I will move into the quality control of vaccines and how to achieve the best sensitivity and specificity in your production. So things to be aware of when you're developing your vaccine is that it's very important that you choose the most prevalent serotypes, especially when you are working with the most critical strategic decisions in your vaccine development and are working with pathogens which has multiple circulating variants. And here we can say it's the same for pneumococcus, dengue, meningococcus, and other bacteria and viruses. So it's very important that you actually go in and look at the surveillance data to choose the most prevalent one, so you can ensure that your vaccine is delivering the maximal public health impact and also demonstrating efficiency and herd protection, and also still keeping and maintaining a relevant and feasible manufacturing and cost of the vaccine. Second, we come into the surveillance data because you cannot choose the most prevalent serotypes if you don't have good surveillance data. So it's also important when we look at the surveillance data, we actually are aware of what methods are being used to produce these surveillance data, because it can be affecting your vaccine development later on. Next is your vaccine development. You have now chosen the most prevalent serotypes, but it's also important that you don't just trust the strains you're buying or acquiring. You need to remember to check that the strains you have acquired also have the right serotypes, or else you can end up with a vaccine which is directed against a wrong serotype and is not showing the effectiveness when it comes to the final testing. So all the early stages of your vaccine development, it's important to check what you get into your house is actually what you expect it to be. And there have been cases around the vaccine development where they actually got a strain which was not the right strain, where it was done by genotypic methods, and it then later on showed that it was not the right serotype. And they then had to go back into the development and replace that one and start from scratch again. So very important, check that the strains you're acquiring is actually what you expect it to be. And then finally, when you come into your quality control, it's also important to find, for the vaccine you're developing, find the right antibodies for your quality control, and also decide on which method is the right method for you to do. Is it nephelometry, or is it another multiplexing method? That's an important point to already in the beginning look into what to do and find the best fit for you for the method. Find the right antibodies for the method you're using. In this presentation, I will focus on these two areas. I will look at how you're coming to the right surveillance data. So when you start developing your vaccine, you also can trust the surveillance data. And then I will go into finding the right antibodies. And here I will focus on the WHO recommended method, which is the nephelometry method, and which antibodies is actually the best for that method. So a little history about pneumococcus, because that's where I started out. That's where my heart is. So back in the 1902, State Serum Institute was founded to produce antiserum, and that's where the name State Serum Institute is coming from. It was founded to produce antisera against infectious diseases for treatment. In 1915 and up to 1945, the structure and the antigenicity of the pneumococcal capsule polysaccharide and its role in human diseases was fully described, so you had a good idea of how the bacteria looked like and how the genes was expressed in the bacteria. In 1934, State Serum Institute started developing monoclonal antisera against the different pneumococcus serotypes, and it was used for serum therapy for infected humans. And because they were producing these antisera for treatment of patients, they actually received the pneumococcus strains from around the whole world to produce these antisera, so they could ship it out to the countries, so they could also use it as medicine against pneumococcus. And by 1945, more than 80 serotypes was actually described within pneumococcus, and antisera was raised against them. In 1951, that was when penicillin was introduced, and that made a swift in the treatment of patients. Now you had an antibiotic, which could be used for the treatment. So that left the monoclonal antisera to be stopped, to be used as a serum therapy. But people still needed to look at the surveillance. They were still interested in seeing which serotypes was out there, so they were still being used for serotyping of pneumococcus, and it's also considered the gold standard method today within serotyping of pneumococcus bacteria. In the 1970s, and actually up to today, the SSI and now SSI Diagnostica Groups and monoclonal antisera is being used in the vaccine industry for quality control of the vaccine in their nephelometry method. So it started early, and we are still in that space and just fine-tuned our antisera to now have a dedicated product for that, which is also being supplied with the image analysis paper or immunochemistry analysis paper for the nephelometry method. The antisera is also being used for assay development for clinical studies by the pharma industry, but also being used for diagnostics methods within the IVD products in life science. So that's a little history about how the antisera came about and how the typing of pneumococcus actually started. And if we then look at surveillance as a general, by the late 90s and early 20th century, it became clear that the pneumococcus bacteria was one of the leading causes of severe illness and death worldwide. And by 1960s and the 1980s, they found out that the antibiotic resistant pneumococcus was emerging globally, and that reinforced that the treatment alone with penicillin and other antibiotics was not sufficient, and prevention was needed. So because we had these monoclonal antisera when using the antisera for treatment, non-reported surveillance data was actually collected already back in the 1940s and up to the 1970s. So a lot of data was already available on pneumococcus. And in the 1990s, national surveillance systems started up in different countries, in different regions, for surveilling the invasive pneumococcal disease. And that established the information which was needed to start the vaccine development. But how is a surveillance system then set up? So I will give you an example of how it functions in Denmark, because that's where I'm from and where I have a good thought, but it's the same all over. But if you look at the surveillance system for pneumococci in Denmark, it starts out with the patient coming into the hospital sick. They will take the clinical laboratories will take the clinical samples, a blood culture, it could be a swab, it could be different things to find out what the patient is sick with, and they will then take the isolate, and if it's a pneumococcus, and for the pneumococcus invasive isolates, it's mandatory to send it into the National Reference Laboratory. They will also share data on which source is the isolate coming from, the patient information on dates, age, and sex, but also if they have been vaccinated or have gotten the spleen removed. That's important information for the surveillance industry to know. So in Denmark, it's Statens Serum Institute, which is the National Reference Laboratory, and they will then start looking into this isolate to confirm it's a pneumococcus. And that is done by using a slightly modified version of the WHO guidance document on how you're doing your phenotypic method of and confirming that you have a pneumococcus bacteria and which serotype it is. So you will first take your isolate, put it out on blood agar plate, where you will identify the alpha hemolytic colonies, which is one of the criteria for a pneumococcus bacteria. Then you will do gram staining, catalase test, and Antigen test. The first thing is if you look at the gram staining, that it has to be a gram-positive Diplococcus or short chains of pneumococcus bacteria, and it has to be catalase negative. If that's confirmed that it's gram-positive bacteria and a catalase test is negative, the next step is to do the Antigen testing. And if you have an Antigen test which shows a diameter of 14 millimeters, then you actually report it as a Streptococcus pneumoniae. If it's below 14 millimeters, you then move on to the next step, which is spill solubility testing. And if the bacteria is spill insoluble, it's considered an alpha hemolytic streptococci other than pneumococcus. And that one will then not be reported in. If it's then spill soluble, it's reported as a pneumococcus positive or streptococcus pneumoniae positive. And you will then move on to your phenotypic method, which is the serotyping of the Streptococcus pneumoniae. So this is the workflow. And when you then have your serotype, the reference laboratory will then report back the data to the hospital on which serotype it is, if it has antibiotic resistance pattern, and other informations they have from the surveillance system on vaccination status and previous infections. They will also report into the local surveillance data, which is MIBA. They will report it in the same data into ECDC, which is the European Center for Disease Prevention and Control. And finally, they will also share the information with WHO. So these data for all the countries who has this surveillance system set up for pneumococcus, they will report it into the authorities or the higher authorities, and these data is then publicly available for the vaccine industry to go in and see which serotypes is then the most prevalent. Of course, it's not all country who has this as a mandatory reporting, but it is growing and more surveillance data is being available out there. So how does this then lead into the vaccine development? So today everybody knows that pneumococcus is a well-known vaccine candidate. And already by 1977, the first pneumococcus vaccine was introduced, which were the polysaccharide vaccine for the adults. And later on in 1983, this vaccine was further developed by Merck to become the 23-valent polysaccharide vaccine, which is well known in the industry. So what we saw out of, we were now protecting the elderly population with the polysaccharide vaccine, but we also we were still not seeing the full effect of the vaccine. So that led to looking at the children, because the children is the carrier of the pneumococcus and are affecting the elderly population. So the first conjugated vaccine for children, the 7-valent, was introduced in 2000, and that one showed a decrease in invasive pneumococcal infections, both in children, but also in the elderly population, because now we actually hit the bacteria from two sides, both the children who were carrying the bacteria and also the infected elderly population who were now getting their own vaccine against the 23-valent serotypes, along with we are limiting the spread from the children. And today we actually have multiple conjugated vaccines available, the 13-valent from Pfizer, the 15-valent from Merck, and the 20 from Pfizer, and 21 from Merck. There's multiple vaccines in the market, also from other companies around the world in India and China. So there's a lot of work being done in this area within pneumococcus, and we're working on getting a better serotype coverage, because there's 92 serotypes of pneumococcus out there, and we want to remove the ones which is most prevalent, or the switching of the serotype after the introduction of the vaccine. So it's still important, even though we have the vaccines, to keep the surveillance ongoing and the reporting on it, because we are seeing that as vaccinations or vaccines is coming out and being more and more used, we see that the serotypes are circulating, and the disease burden, we want to make sure it's always kept as a minimum. And if you don't do your surveillance, you will not be able to detect new emerging serotypes coming up, and you cannot ensure that the vaccine remains effective. So this is really important that we keep the surveillance data coming in and doing the surveillance of the disease, so the vaccine industry can then develop the right vaccines for the right market. Because it's also important to look at the different markets, because we have different serotypes, which is the most prevalent in our different regions of the world. Then I will move into the phenotypic way. So why phenotypic is critical for your vaccine development. And here I'm going to give you a short introduction to the two main methods being used around the world within the surveillance industry. And you have the phenotypic method for pneumococcus. Back in the 19th, Frederick Neufeld discovered the Quilling reaction, which is a microscopic method. And it's actually the one considered today to be the golden standard method for phenotypic typing of pneumococcus. And it needs a whole bacteria to perform the test. And you will look in the microscope, you will detect the bacteria and an antisera, mix it together, and if it's by example a type 1 capsule or bacteria and a type 1 antisera, you will see the capsule of the bacteria swell up. And that is something you can see by the eye in the microscope. So that's the method, and the golden method today still is to look in the microscope for that capsule swelling reaction. Then you have the genotyping method, which is the new emerging method being more and more commonly used. It started out in the 90s, which is a PCR-based method. It's a good complement to the phenotypic method. It requires bioinformatics skills. And here you are looking at the bacterial DNA. So you, of course, need to have a DNA extraction system, a thermocycler, and a sequencing platform to perform the test. So that's just shortly on the two different methods being used in the surveillance industry. And then if we then go in and look a little deeper on the two methods, so genotypic methods are detecting the capture of polysaccharide genes, where the phenotypic method is actually detecting the expressed capture of polysaccharides on the surface. So genotypically, the genetic potential is shown, the DNA sequence, and the potential functions of the bacteria. So everything is what you have in your gene, and the vaccine relevance for that is indirect, as it can predict serotype potential. But where the phenotypic method is actually showing the actively expressed genes and the observed characteristics and behavior exhibit under the specific environmental conditions. And here the vaccine relevance is that it's a direct impact because it's what the antibody will actually bind to. And it's being used and very important when you do OPA testing in your vaccine development, because you want to see that antibodies you produce with your vaccine actually can bind to this expressed genes and show a killing effect within the OPA assay. But what is the strength about the two methods? So within the genotypic method, it has a high sensitivity and specificity. It's not dependent on how well the bacteria grows on a plate, and it enables the lineage tracking and surveillance. It detects the silent and also the low expressed genes in the bacteria. It's highly scalable and high throughput, and it's able to guide us to look for new potential phenotyping, serotypes within the bacteria. The phenotypic method strengths are that it can be measured biological relevant expression. It directly reflects the immune recognition. It captures the regulations, the phase invasion, and the environment. So everything which is affecting the bacteria is actually shown in this space. It's the golden standard for serotyping of pneumococci, and it's clinical relevant. So that's the strength of the two methods. If we then look at the weaknesses of it, so for genotypic methods, it doesn't guarantee the expression of the gene. It cannot detect the regulatory or functional defects reliable, and the vaccine relevance must be inferred. It requires specialized skills and tools and equipment to do it, where the phenotypic method is, the weaknesses of that is that the sensitivity to growth conditions and the operator's skill to do the testing in the microscope. It has a lower throughput. It's subjective interpretation, so if you're not good at looking in the microscope, it can be hard to interpret it. And some serotypes can be viable and transition during your growth patterns. So, but why is it then the phenotype is critical during your vaccine development? And this is lessons learned from pneumococcus. So the vaccine targets expressed genes. So it's expressing the capture of polysaccharides and not the potential genes in the bacteria. The risk when relying only on a genotype, it's increased the vaccine escape. The genetic potential to express a vaccine capsule does not always translate into what is actually an effective antibody recognition and have the killing effect as the phenotype method can show. So the phenotypic expression, not just a gene which is present, but it's actually the expressed genes is ultimate for determining your protection of the vaccine. But a combined approach benefits the research. So we still need to remember those methods because it will be important for the research and the surveillance industry. So by using both phenotypic method and genotypic methods, that will improve the research accuracy on surveillance and also leading to a better outcome and understanding of the bacteria and how we can then look for new serotypes. And you can say this has been the approach within Pneumococcus for many years, because here you already have the polyclonal antisera for phenotypic method available, so it makes it easy to do your phenotypic method along with your genotypic method. But what happened when we look into new and emerging areas? And here I chose to give an example within Klebsiella pneumoniae, which is a new potential prioritized vaccine candidate. So WHO actually have looked into this and seen that the cavanase-resistant Klebsiella pneumoniae is currently the most dangerous antibiotic-resistant threat to the global public health. And that's why it's now being prioritized as a new vaccine candidate. But how to decide which K types to direct your vaccines against? That's hard because the available surveillance data are minimal. And if you look at the genotypic data, these should be defined as capsular locus with a KL for genotype to express that this is a genotype. The way the bacteria was detected was with genotypic methods. And the phenotypic data should be defined with K as capsular for the serotype. But in practice, what happens? Because there's no commercially available monoclonal antisera in the market, genotypic data is using a mixed nomenclature and now including both the KL and the K types. And back in 2024, this actually led to a new symposium being held within the Klebsiella space. Because if you're starting to look at it from a vaccine perspective and a surveillance perspective, you need to have a place where people could come together and discuss these very important topics. And during this symposium, which was held in the Institute of Pasteur in Paris back in 2024, a publication was made by State and Serum Institute. It was funded by Bill Gates. It was a collaboration across Australia and Denmark to look at the genotypic method and the phenotypic method and look at the Klebsiella bacteria. And SSI Diagnostica Group was part of that project because we were developing the antisera against the Klebsiella K types. So this was a large genomic and phenotypic study on Klebsiella pneumonia, which was presented at this first symposium. The study contained data from 299 clinical samples, which was collected around the world. The method used was the genotypic method with the whole genome sequencing. And the phenotypic method was the golden standard, which is our immunoelectrophoresis and counter-electrophoresis assay where you use monoclonal antisera. This is the golden standard method, which has been used in the past in the surveillance industry. And the outcome of this study showed that when you did your genotypic method, you've had 171 of them, which was known K types. They saw that 54 types was new genotypic serotypes. And then they also had 70 other genotypic types, which was detected. But when they then did the phenotypic method, they actually found out that only 127 of these 171 was actually K types within 1 to 99. They found out that 16 of them was actually other K types, and 28 of them was not untypable with the current antisera present. So these could be potential new serotypes. They also found out that 50 out of the 54 samples was actually known K types, so it was not new serotypes, which the genotypic method found. It was actually known types. Only 4 of them was actually untypable and was looked at as new serotypes. If they looked at the other genotypic serotypes, they also found out 30 of them was actually known K types, and 40 of them was only new types. So this led to that, that 80% or 41% of these Klebsiella serotypes was actually mistyped by genotypic method. So this shows the importance of the vaccines or the surveillance data being done phenotypically and not just genotypically, because phenotypic is the expressed genes and what we want the vaccines to be directed against, or else we will be developing vaccines to something which is not correct. And you can say, if you looked at out of the 299 clinical samples, 80 samples was mistyped to be a new genotype. So that was also quite a large pool of the types which was mistyped. And this is the discussion of the poster, where they actually saw that some of these vaccines which is being developed actually was known K types and not new serotypes. And you can say they discussed that it is important when you're doing it and you're recognizing new genotypes that you actually also check that it's actually new capsular types and it's expressed in the genes. And you can say during that vaccine symposium, vaccine candidate was presented by different companies, and a lot of them had included the KL102 to be a new serotype, which they would put into the vaccine because it was much more prevalent than the other ones. So they were looking at the KL102, which was a genotype, but was it actually a new genotype or phenotype? And what it actually showed in this study was that it was K18, which was already well defined within the phenotypic method, and your vaccine now had wrong data being produced because they were expecting it to be KL102, but it was K18. So is this now still the most prevalent one, or was there other serotypes which was more relevant to put into your vaccine? So the risk of reducing the specificity when your vaccines are designed and to cover multiple and overlapping K types. This is the importance of looking at it from a phenotypic method compared to the genotypic method. And that was actually also the conclusion by the authors of the poster, was that in conclusion, it's important to incorporate both genotypic and phenotypic characteristics to make sure you identify the novel capsular types. So, but I also want to highlight again using phenotypic method and genotypic method, that is what will improve our research accuracy in surveillance, because we need to find out if new emerging serotypes is coming. But again, we need to understand how it will impact the vaccine industry if we are not using both methods and actually looking at the expressed genes. So now I want to move into when you're actually doing your vaccine development and you want to achieve the best sensitivity and specificity in your vaccine QC in the nephelometry method. So we did a massive study because there's been so many discussion. Is monoclonal the right antibody? Is monoclonal the right antibody? And as a scientist, I want to understand and I want to see which method or antibody is the right antibody for this method. So we were using the rate nephelometry method, which is a immunochemistry method, which is measuring the light scattering from antigen and antibody complex. It enables the quantitatively determination of the polysaccharide, the capture of polysaccharide content. It's widely used for polysaccharide and conjugate vaccines. It's suitable for lot release and stability testing. It has a high precision and a throughput compared with classical immunodeficiency assays, and it's accepted by WHO and NARs when validating and showing fit for purpose. And it's actually one of the recommended methods by WHO in the guidance for quality and safety and efficiency of conjugated vaccine. So just to shortly show the method, so you will have, when you have your sample, it will scatter the light through it, and depending on how big your complex is, how many antibodies and antigens, which is bind together, the scatter of the light will then be recorded and give a instrument response. The rationale behind the study and the design was to find the suitable pneumococcal monoclonal antibodies for certain serotypes, compare the performance of the monoclonal antibodies up against the polyclonal antibodies. We looked at material from NIPSC and the high titer monoclonal antisera from SSI Diagnostica Group. We used the same protocol for both antibodies. The dilution of the antibodies was based on the SSI Diagnostica high titer antisera and adjusted if necessary. And we did 7 different validation steps on each antibody in the nephelometry method to see which was performing the best. So this was the validation and the acceptance criteria of the method. So we were doing a standard curve where we were looking at the R value, and we were looking at the CV value between the different reading points where we were doing triple determinations. We looked at reproducibility of the standard curve. So, and here the acceptance criteria that the CV should be below 20%. We also had a blank sample with us to actually have a cutoff for the further testing. So we were saying that the blank had to be below half of the 0.5 microgram per ml reading on the standard curve. We ran a positive control of 2 microgram per ml, and it could have a variation of 20%. We also tested 24 cross reactions in a concentration of 4 micrograms per ml against each antibody, and again using the same acceptance criteria as we had for the blank. We did a pool of the 23-valent vaccine where it looked like the vaccine, so 50 micrograms of each serotype was present in this pool, and it could have a variation of 30%. And then finally, we did a 13-valent pool symbolizing the 13-valent vaccine. with the same content as the vaccine. And again, it could vary with the 20%. So this is the material which was being used, the different clones from NISPs and the SSI Diagnostica polyclonal antibodies. So all of these clones and high titer antisera was used for the testing. So first we looked at the standard curve validation, and already here we saw that some of the antibodies was not performing. We had the SSI Diagnostica polyclonal antibodies, which were all fit for purpose and passed the standard curve within the acceptance criteria. But with the monoclonal antibodies, what we saw was that 42% of them were not able to produce stable standard curves. So all the pink serotypes you see here gave us issues in the nephelometry method. Here's an example of serotype 7, where we actually saw that this was the monoclonal antibody from SSI Diagnostica, where we had a nice straight standard curve, but the antibody from NIPS was actually not fit for purpose with the polysaccharides from ATCC. Of the ones which passed, here's an example of type 1, where we actually saw the exact same curve for the monoclonal antibody and the monoclonal antibody. So some of them worked, and that's the ones coming out with green. For some, we had to do a second dilution step to get it to fit, and that was the 17F. So that one required a less diluted sample. Of course, we also tested all the other serotypes to see if it was the dilution which was not good enough, if we needed to use it undiluted or more diluted. But these 8 serotypes was not fit for purpose. They could not be used in the rate nephelometry method. Then we looked at the reproducibility. We, of course, just took the one which passed the first step and put it over to the reproducibility testing. And here we also saw that 3 of the serotypes was not able to be reproduced. We had a large scattering of the standard curve. We tested it over multiple days. We tried new dilutions, still not. We didn't saw any stable data, and were not able to reproduce the data for example, here 17F. Unfortunately, we had to leave out the 19F after this testing because we didn't have enough material and we could not get the material home in time to include it in the rest of the testing. But after this reproducibility testing, we lost 3 serotypes. So now with the 10 serotypes left, we moved it on to the positive control, and we wanted to be able to see if we could refine the 2 micrograms per ml using the standard curve calculations. And here we saw that all of them passed. All 10 passed this part. But of course, we had the non-approved, which were not passing it. We actually tested them just to see if we could see them again. So all the 10 left monoclonal left passed this acceptance criteria. Then we looked at the cross reactions, and here it was a little hard because the monoclonal antibody was not specific. It was a 6A and 6C antibody, so it was not specific. And that's also what we see in the testing, that it actually had small cross reactions to 6C, serotype 6C. But of course, we looked at it from a 6A perspective, so it passed this cross reaction criteria. But 10A and 20 actually showed cross reactions and were not specific for 10A and 20. So those 2 was taken out of the pool, and only 8 was left for the further testing. When we tested the 23-valent pool, all SSI Diagnostica antisera passed this step. And as 6A is not part of it, we only tested the 7 out of the 8 approved monoclonal, and the 7 serotypes actually passed this step with the 23-valent pool. We tested the 13-valent pool, where we included the 6A. And here what we saw was that with the current setup where we run the pool undiluted, we had 5 of them which didn't pass that testing, but with a further dilution of the pool to 1 to 2, then they could be used. So if we adjusted the assay and diluted the 13-valent pool even more, then it could be used for the testing with the monoclonal antibodies. So to summarize this up, what we saw was that there's the monoclonal antibody, which is detecting multiple epitopes on the serotypes, all past this study. But 63% of the serotypes of the monoclonal antibodies didn't pass this testing and could not be used in the same fit purpose in the nephelometry as the monoclonal antisera could do. We actually only had 2 monoclonal antisera passing the whole testing, the whole protocol. 25% of them had to be adjusted to fit the protocol and pass the protocol. And then we had, of course, that one serotype which we unfortunately had to leave out because of lack of material. So this was a really comprehensive study where we looked at the monoclonal, because I'm not in favor of one or the other, but I know within Pneumococcus, monoclonal antisera is better and more specific and sensitive because it can hit multiple targets on the polysaccharide, where the monoclonal is only hitting one target. So that was what we also saw in this study, and what I wanted to see if it was the truth. So after this, we actually wanted to show why there is a difference between the monoclonal and the monoclonal antibodies. And I have a video here I want to show you. So that was just to summarize up why we are seeing that difference between the monoclonal and the monoclonal antibodies, and also references of why we are seeing the differences. So just to summarize up why it's important to look at the whole picture when you're doing the vaccine development in a summary. So for the for Klebsiella, Pneumococcus, and other bacteria, we start looking at the infection. That's where everything starts. Then you have the diagnostics, and here it's important that you have good diagnostics to detect the bacteria or detect the infection. And then, of course, the surveillance industry should always screen with both the genotypic method and the phenotypic method to see what serotypes is available. And those data will then lead into the whole surveillance industry. And it's important we don't use only the genotypic method, but we actually check that the genotypic method is aligned with the phenotypic method, or if it's a new potential serotype. I wouldn't recommend using monoclonal for the surveillance industry because then we're only targeting one epitope, and that can mislead which genes is actually expressed, as some of them will express multiple epitopes. So it's important when we look at new phenotypic or serotypes that we try to make monoclonal antibodies to actually induce a true immune response. And if you do that, you will have the best data for your vaccine. So that was the presentation, and I hope you enjoyed it and got a little round trip around the world of both the surveillance industry, but also how you can use it into your vaccine development. Thank you for listening. And
Thank
I'm
you.
now open for questions. I know I hit the target on the time exactly.
It's okay. Thank you very much for your presentation. That's really good, and also you also, cover a lot and share a lot of knowledge with us. So, okay, I see our questions. Any questions from audience, please?
I would stop sharing, but you have my contact details here if needed
Yeah,
and
yeah,
want to hear more.
yeah. Thank you. Um, Any questions? Oh, I saw there is a questions from the chat. The questions are from Samimo. And the question is sometimes some high tari antiserum meet all acceptance criteria for 1, 2, 4, and 8 dilution, but samples meet acceptance criteria 4. So how can we explain this?
Yeah, you can say when we are developing the antisera for the purpose, we are using the ATCC capsules. And of course, they will not always look the same as your own polysaccharide development because the way you purify your antigens can differentiate between all the vaccine companies. But what we did was to choose some commercially available polysaccharides. So we chose the ones from ATC, which we are hitting it up against to ensure that we can show the right standard curve and that it's actually fit for purpose. But of course, depending on how good your purification is, and the strains you're actually using for your vaccine development, they are expressing the genes differently. So that is why you can see variance in the dilutions. So we only give a recommended dilution. It can maybe mean that you can do other dilutions for your vaccine, depending on how strong and good your antigen is.
Thank you for your answer. Yeah. Any other questions from audience? Okay. Actually, I have a questions for my self. I'm very interested in surveillance system data. I saw that in your presentation you mentioned about that can because the projects that you are working on is our collaboration projects, right? So you mentioned about the data sharing for the surveillance system data, and they share it with MiBi and European centers and also WHO. So I'm wondering how you share that kind of data source system, or it also accessible from public or only used being for you, your collaborators.
You can say, depending on which country you're in, it
Mm,
differentiates. So you can say in Denmark, it's the National Reference Laboratory who has access to put in the data into the survey into MiBa and ECDC and WHO for the DK's data. For the UK, it's the public health who will do
mm,
that reporting. But the data is actually publicly available when it's first reported in. You can look into the WHO webpage and get all the surveillance data out of that. And the same goes for your national data and also the European or the American surveillance data.
okay, okay,
So they are publicly available, and you can actually get reports out on an annual basis how the serotypes have differentiated and where we are seeing the different serotypes popping up.
mm. I think that's a very good news for us because usually, because I'm doing the research work on AI-enabled supply chain resilience, so I that kind of data will help me to for my work. Sorry, I have another questions.
Yeah.
Yeah. During your experience working in vaccine development, and also the nowadays the AI is very popular. Have you seen any cases that use the AI to support vaccine development, and also any opportunities that you say in the future to use AI support vaccine development, or any challenges that you have seen that in that area?
You can say I'm a big fan of AI.
Yeah, me too.
It's a great support,
Mm,
and it's a good place to actually get it to support you on actually combining the surveillance data and also differentiating it into the different regions of the world, how the surveillance data uh, is divided.
mm,
But I will say it's always important to check your references in the AI tool you're using
yes.
because some of them are not relevant, but you
Mm,
can actually ask it to only use the the WHO data or the local data for a country. So just be aware of which reference is being used. But it can also be used to look at the carriage, um, especially within Pneumococcus. So you can get it to support you on finding all the right publications to look further into. So I'm using it as a tool to give me a summary
yeah,
of all the publication and which one I need to dig deeper on. But I
yeah,
always check my references because I have seen that sometimes the references is not the best. So it's
yes,
important
yes.
to always check that the AI tool is also giving you the best surveillance or references needed. So
Mm,
so be mindful when using it, but use it because it will
yes,
definitely remove a lot of burden in your work
yeah, yeah.
because
That's
it's
definitely.
it's combining all the data for you
Yeah.
instead of you needing to do it.
Yeah, thank you. To summarize that, based on your answer, yeah, so the AI can support to understand the surveillance data, and then you can use that for your vaccine development. And the other hand is like to um, um, summarize all important information or documents or reports, and meantime also manually check whether it's correct, right?
Yes.
Okay.
Yeah,
Yeah.
and
Thank you
it
very
can
much.
also be used in your vaccine development on looking into which other vaccines is under development. Of course, it will not tell early stages, but you
Yeah.
can also see what is actually other companies working on as soon as they get into clinical.org. then you actually can have it look into that and also give a summary of what is it being seen, which publications is out there with issues, what
Mm,
have been seen in previous vaccine studies. So you actually can also ensure that you don't do the same mistakes which have been done in the past.
yes, yes. Okay, thank you. Thank you very
Thank
much
you.
for your presentation.
Yeah,
It's very valuable for us, and yeah, hopefully we can collaborate in the future in some way. Yeah, because of time, so I will close this seminar. So thank you very much again. Yeah, have a good
and
day. Yeah,
yeah, and thank you for having me. Thank you for
thank
listening
you.
in to
Yeah,
everybody. Okay, thank
bye.
you. Bye.
Take care. Yeah.